You have a donor and you have a recipient of the bone marrow from the donor. And after the transplant is done, you can use these procedures to follow whether or not the transplant has been effective by looking at the blood cells in the recipient and saying, is the original genetic source of those cells those of the recipient, which you're hoping it won't be, or those of the blood marrow donor, and you're hoping that it will be that. And you can track the progress of the transplant using the very same procedures that are used for paternity or identification testing.
As far as disease diagnosis, if I can return to that, is this RFLP procedure then used to determine, for instance, whether an individual suffers from cystic fibrosis?
It can be. There are now easier tests than the RFLP procedure, but that was certainly early on the procedure in the research stages that would have been used.
You also brought up the use of this technique, for instance, in the poaching scenario. Is this RFLP technique also used to help save endangered animals?
Yes.
What about this technique, this RFLP technique, and for instance, plants? Does it have any role there?
There are, the main role that I'm aware of, and I'm not a plant biologist, so there may be many other aspects that it's useful for that I'm not aware of, but in plant breeding, where a company has a corn hybrid that has specific characteristics, those characteristics can be identified and tracked in that hybrid using RFLP analysis with exactly the same types of procedures that are used in testing for paternity.
Does this RFLP testing technique play any role in, for instance, identifying the remains of American war dead?
It has, yes.
And does that include, for instance, remains from soldiers killed in battle?
Yes.
Now, you've described a number of general areas, including human identification, that this RFLP technique is used. Would the human identification use of this technique be the majority or most or a small portion or what?
Overall, it would be a small portion. If you look at anyone in science who might use this procedure or this set of procedures that makes up RFLP analysis, human identification would be a very, very small fraction of its total application.
Is this a technique that was basically born out of forensic science, or did forensic science borrow it from the greater scientific community?
It would be that forensic science borrowed it from the research community.
What about in terms of how wide this technique is used? And I'm talking about geographically. Is it used in the United States and England alone, or is it used elsewhere?
Sustain, rephrase the question.
As far as the RFLP typing approach, is it used strictly in English-speaking countries, for instance, or is it used in some wider area than that?
No, it's used in a much wider area than that.
And would that include, for instance, in countries around the world?
Yes.
Now, if I can return to the RFLP method, first of all, would it assist if I asked you to describe briefly this method to use, again, the drawing pad for purposes of demonstrating what happens?
I probably can't do it without scribbling on the pad.
Okay, fair enough. Marked as People's 241.
241. We'll call this RFLP drawing. Mr. Clarke.
Thank you. In fact, Dr. Cotton, if we could, before we do that, could we go back and perhaps if you could step down? I'm going to ask you to put just very brief titles on the previous exhibits, the previous drawings. Perhaps we could start with the 46 chromosomes chart that I believe is People's Exhibit 236. No, 235, excuse me. That's labeled 46 chromosomes. Would that be an appropriate title for that chart?
It will do.
Okay, perhaps you could just underline it, and then we'll underline the titles on each one. And
All right,
then-
235 will be labeled in chromosomes, underlined, 46 chromosomes, underlined.
All right, and if you could go to the next drawing that you made. Which is Exhibit 236. Does that drawing depict on the left the single pair of chromosomes and then the two strands of DNA to the right?
Yes.
What would be a short title for that drawing?
Why don't we just put chromosome pair?
Okay.
Does it matter where?
Why don't you put it in, if you have a different color, I think you could put it in the middle, it would be fine, as long as you underline it. And then as far as the two strands of DNA to the right, would it be appropriate to give that a title?
No, that's just the two strands
All
from
right.
these two chromosomes, so that's still a chromosome pair.
Okay, if you would, go to the next diagram, which I think has already been titled double-stranded. And would that be an appropriate title for what I believe to be Peoples 238?
Sure.
Okay, and that just leaves us then the next page.
Let's add DNA.
Okay, and then if you would go to 239, the last drawing. And is that a demonstration of single-strand DNA?
Yes.
Does your T have a cross on it? And again, Your Honor, that would be People's Exhibit 239. Now, Dr. Cotton, let's provide you with a new page. Which will be People's 241, I believe. First of all, why don't we label it at the top, RFLP method? And if you would now use that particular piece of paper to help describe this RFLP testing process.
Okay.
Go ahead.
No, we just. What I want to do is go back to the idea that you have two chromosomes, or you have a chromosome pair, and that we're going to look at the DNA from that specific chromosome pair. And I want to explain to you what we keep talking about looking at differences in the DNA. I want to show you an example of the kind of difference that we're looking at, because I think that's the only way it will really make any sense. And then, once we've done that, we can sort of walk through the procedure.
All right.
So let's go back and say we have our two chromosomes worth of DNA here. And they have, you know, the A and T and GC base pairs all along. And so if we're looking around on these chromosomes, you will come to a section that contains what is referred to as repeated sequences. And I'm going to make a very short and short meaning short in physical length, simple example of this, but this is not an unrealistic example. Suppose we come along and we're looking down the sequences of bases, and we see that we have a C followed by an A followed by a T. with another C followed by another A followed by another T, and another C followed by another A followed by another T. Now, what I'm going to do is only draw one side, so if you'll just assume with me that the other side has the appropriate matching base. So we have this sequence CAT repeated 3 times. Now, if we went and looked at the other chromosome, this is now, from one person, and you're going down the chromosome and you're looking at the base pairs, and you come to sort of a matching section, and you find that it has a C followed by an A followed by a T, and you see that that sequence of 3 base pairs is repeated quite a number of times. But on this chromosome, it's repeated more times. Here we have, on the one on the left, we had 3 repeats. And here we have, on the one on the right, we have 5 repeats. So we have this short section of something that's repeated over and over again. Now, you have to sort of think about if we made this same drawing for yet another individual, and another, and another. What you would find is that another individual might have a section, they'd have this, you go on the chromosome, they'd have this same repeat, CAT, but it might be repeated 10 times on one of their chromosomes, and 18 times on the other. So that everybody has the CAT sequence on their chromosome, but not everybody has the same number of repeats. That is the difference that RFLP testing looks at. So if you imagine in your head that we had a method to cut out this section, and then you add to that we had a method to measure how long this section was that we cut out. For this person, if we could do that measurement, we'd have a section that was 9 base pairs long. And for their other chromosome, we'd have a section that was 15 base pairs long. So this one becomes 9, and I'm going to use the abbreviation BP to stand for base pairs. Base pairs being the GC or the AT pairs. And over here, our 5 repeats is equal to 15 base pairs. So, and then if we had another person, and we could make another diagram just like this, if they had a chromosome that had 10 repeats, that would be a section that was 30 base pairs. So, it's those lengths that are different from person to person. And in this abbreviation, the P standing for polymorphism, the L stands for length. This test looks at length differences in the DNA, and the length differences are created by the number of repeats that happen to be end-to-end at a specific location on a chromosome pair.
So is it then the case that this RFLP method is simply a tool to determine how we vary from one another as human beings at this particular region of DNA where these repeats vary?
Yes.
As far as the method itself, to your knowledge, do we have a prepared chart that illustrates how this difference is actually determined using the RFLP test?
We have, you have, and I've looked at, a prepared chart that doesn't illustrate this, but illustrates the set of procedures that allows you to ultimately look at these length differences.
And is it these length differences that are simply what you're looking for as a DNA scientist when you're using this RFLP method for identification?
That's right.
All right. And Your Honor, with the court's permission, I'd ask be marked as, I believe, People's Exhibit 242,
People's 242.
a fairly large repair diagram entitled, I believe, the RFLP method. Now, Dr. Cotton, first of all, as you've described on your drawing, People's Exhibit 241, where you've described this differences amongst us as human beings in the number of times we repeat a particular sequence, how are these particular regions of DNA picked out or selected to use for identification purposes? Does that question make sense?
It makes perfect sense, but it takes that whole procedure to do that.
Okay, all right, then why don't we go ahead and if you would, then describe this RFLP method in the basic steps that are delineated, and perhaps I can ask a question first. Have you had an opportunity to look at this large chart entitled the RFLP method that was previously prepared before coming to court today?
Yes, I have.
And in that review, does this diagram basically describe the individual steps that are part of this RFLP DNA typing
Clarke,
method?
Excuse me, Mr. Clarke, let me interrupt you just a moment. Let me see counsel without the court reporter, please. Thank you, counsel.
All right, Dr. Cotton, with regard to this diagram then, could you describe, would utilize this diagram and the drawing you've previously made to describe how this RFLP typing method actually works?
The diagram on the board has everything numbered, so I'll try to relate what I'm saying to the number. So let's say you have a sample, some biological sample. The diagram illustrates either a tube of blood or a blood stain, but this could be, as I said before, a hair root. It could be a semen stain. We'll just say we have some biological sample. And the first thing that happens is that the DNA is extracted from this sample. What that means is the DNA is purified from the other components of the sample that you don't care about. The DNA is in the cell and you have all these proteins. Proteins are nice, but for purposes of DNA testing, we don't need them. So you're purifying the DNA away from the other cellular components, which include proteins and cell membranes and things like that. That's step number one.
All right.
Step number two on the chart says the DNA is cut into fragments by a restriction enzyme. When I said a little while ago that if you had a method of cutting out these sections where the repeat exists, that you could look at how long it was. The method is to use a protein that is called a restriction enzyme. And these enzymes have the characteristic that they can locate a specific short set of base pairs. Experimentally, it's predetermined there are many restriction enzymes. There are over 100 different restriction enzymes. And let me just give you an example so it doesn't sound so Greeky. Let's just say we have a double-stranded piece of DNA, and you have this series of bases, C, C, G, G, and you go down the DNA and you find another set where it happens to have C, C, G, G. If you have a restriction enzyme that recognizes this site, it would make a cut wherever it happened to see a CCGG. And I'm just giving you one example. There are many others. So,
Actually, while you're on that drawing, Dr. Cotton, perhaps you can label that restriction enzyme?
well,
Would that be an appropriate description or
let's
not?
call it this is just an example of a restriction enzyme cutting site.
And Your Honor, could that be marked Peoples 243?
243.
Thank you. All right, continue if you would, Dr. Cotton.
I'm going to go back to the previous diagram. The test is set up so that you are using a specific restriction enzyme that we know will cut outside the area of the repeats. So the enzyme that's used in the test is designed so it will cut at the beginning, just before the beginning of the repeat, and just after the repeat, or somewhere close to that. So you purify your DNA, you add your restriction enzyme, and you make these cuts. Now the enzyme cuts many other places, so basically if you go back to the spool of thread analogy, and you've rolled out your spool of thread and it's very long, you've now taken a pair of scissors and cut it up into a whole bunch of small pieces. And among those small pieces are these two that we're interested in.
All right, what step do you take next to locate those interested fragments?
The next step, actually the next series of steps, are the steps that allow you to locate those fragments. So the first thing you do, now that you've got all these little fragments, is that you use a procedure that's called electrophoresis. It has many other uses. You can do electrophoresis with proteins, but in this case we're doing electrophoresis with DNA. And that electrophoresis will allow you to separate out the pieces according to how big they are. So if I had a test tube full of pieces of DNA that were all different size, at the end of the electrophoresis, it's as if I had been able to reach in and put the small pieces down here and the medium-sized pieces over here and the larger pieces down at that end.
If this jury had heard previous testimony about the use of electrophoresis to type proteins such as PGM and EAP, would this be the same tool that you're talking about in the RFLP typing process?
It's very similar, but it's not exactly the same.
All right, go ahead.
The picture on the diagram that's numbered 3 is the illustration of separation of the DNA fragments on an agarose gel. that illustration is pretty okay, but let me try my own and see if I can make it a little more graphic. The gel is about the size of a little smaller than an 8 by 10 piece of paper. Actually, it's about the size of your notebooks, a little bit wider and about that long. It's about 1/4 of an inch thick, and it's made of agarose. Agarose comes as a powder, and you boil it up sort of like Jell-O, and it goes into solution, and when you pour it into a tray, it solidifies. So it has some of the same characteristics of gelatin, although it is not gelatin. So you're pouring this gel into a tray, and if I look at that tray from the side, after I've poured the gel, this is about 1/4 inch thick here. I also use a small mold to make an indentation at one end. If I take that same gel and I draw it as if it was sitting on the countertop where I could look down on it, I would have a series of these rectangular indentations across the top of the gel. Now on the diagram they're sort of showing you where 2 samples have gone in that gel, so the red lines, the red lanes are sort of one that represents one indentation where you could put a sample. Anyway, you have your DNA sample, and the sample is placed into this indentation in the gel, which is called a sample well. And the DNA moves through the gel. The DNA has a charge. It carries a slight negative charge, so if there's a positive electrode at this end and a negative electrode at this end, DNA is forced to move through the gel, and the gel acts like a sieve. Short segments of DNA can move through the gel rapidly. They can work their way through this sieve pushed along by the electric current. And longer, so shorter pieces will move through the gel the farthest. The longer strands of DNA cannot move as rapidly through the gel, and they do not go as far. So this is the way of taking these 2 pieces of DNA. These are now the pieces that we're interested in. Remember, they're part of the group that we're looking at. We have these 2 pieces in our mass of bits. We want to see where they are. This piece is shorter than this piece. So in the gel, the shorter piece, let's say, moves this far, and the other piece cannot move as far. So now these 2 pieces are separated. The piece that was only 9 base pairs is in a different position than the piece that was 15 base pairs.
All right, could we, with this diagram that you've just drawn, would electrophoresis be an appropriate title?
Sure.
And I believe that would be People's 244, Your Honor.
Yeah. All right, Mr. Clarke, I need to allow the jurors to take a comfort break at this point. Ladies and gentlemen, I forgot to mention to you this morning, I don't know if the bailiffs mentioned to you that we have a modified court session today. I have a funeral to attend this afternoon, so we're going to go through until 1 o'clock and then break at 1 o'clock. So we'll take a 10-minute comfort break at this point, and we'll resume again at 12 sharp. All right, please remember my admonitions to you.
Of all, and I'd like to direct you specifically to what's labeled number 3, low gel, and the photograph above that. Does that appear to be a gel?
Yes.
All right. Is that the same, different, or similar from the gels that you're describing as part of the RFLP process?
Well, I'm sure it's different. I'm not exactly sure what kind of gel that is. If it was the RFLP process, and that looks to me like a glass plate, although I can't tell that well. If it was the RFLP process, it would be a plastic tray that has sort of like pouring something into a square or rectangular cake pan, but the edges of the tray are maybe about 3/4 of an inch high.
Okay. Then, taking you on to step 4 on this diagram, People's Exhibit 215, it's labeled for electrophoresis. Is that the same or similar to the electrophoresis process you've described in the RFLP method?
The tank that's shown in photograph 4 doesn't look very different from the type of tank we use. It has space at either end where buffer or some kind of a wick goes, has a plastic lid that comes down to protect the user. What's really different here is that if you look at photograph number 3, where it says load gel, first of all, the position where the samples are loaded on an RFLP gel is very close to the top, more like what I've drawn in this diagram. Whereas there, it's maybe 1/4 of a way down the gel. And these are, in the RFLP procedure, these are liquid samples that are loaded using a pipetter with a disposable plastic tip.
Okay. And then lastly, on this Exhibit 215, where it's labeled running the gel, do you in fact run gels during the RFLP method typing process?
Yes, it shows there a series of gels and 2 power supplies up on a shelf, and we have a similar looking power supply that's connected to the gel tank such that you have a negative and a positive electrode, which is creating the current, the electric current, which is pushing these DNA fragments along through the gel.
All right. Your Honor, with the court's permission, could 215 just be brought over towards this side so the remaining jury members could have a view of it?
Yes, Mr. Chartwell?
Now, Dr. Cotton, at the time the electrophoresis, or step 3 on People's Exhibit 242 is completed, can you see these different fragments on the gel?
No, you can't see.
What's the next step in the process?
The next step in the process is to create a permanent record of the separation of the DNA fragments in the gel. If you tried to pick up an agarose gel with your hands, it would be like trying to pick up a 1/4 inch slab of Jell-O. It would break apart in your fingers. So whereas all the information you want is in this gel, you have to have a permanent way of storing this information. Can I backtrack for 1 second?
Sure.
And then start a new diagram?
Yes.
Let's still consider this 2 chromosome or the 2 DNA pieces from the diagram a few places back. Let me make sure that I straighten one thing out and then go on. I talked about a fragment of DNA that was 9 base pairs and another fragment that was 15. Let's put this more into reality for this test. The sizes we might look at, you could look at a size that was 900 base pairs and a size that was 1500 base pairs. This is more like the size range, and actually you can look considerably larger than this, but this is more like the size range of pieces that you would be looking at in this test. Still, due where the differences in size would due to the different number of repeats in that section of DNA. So that if we go back, we have our 900 base pair piece has moved further down through the gel, and the 1500 base pair piece has not moved as far. When you have this separation in the gel, this is the information that you really want. And the entire rest of the procedure are the set of steps that allow you to visualize where these pieces of DNA went. So if you understand that where the DNA came to in the gel is a measure, an indirect measure of its size, that is, this distance traveled is an indirect measure of the size. The further the distance traveled, the smaller the size piece of DNA. The shorter the distance traveled, the larger the size is. This is the information you're after, and the entire rest of the procedure is how do you see this visually.
All right, Your Honor, for the record, the witness has been referring and writing further on People's Exhibit 244.
Yes.
All right, Dr. Cotton, then would you be, or are we proceeding to step 4 on the diagram, the RFLP method?
Yes.
And could you describe that, please?
Yes, and I won't, actually, maybe I won't draw another one because this is pretty nice here on step 4. What you want to do now is keep this record, or make a permanent record of where these DNA fragments are. And the way that's done is to transfer the DNA in the same relative positions from the gel onto a nylon membrane. And that step, or that process, is illustrated in step 4.
All right, would a laser light pointer help you with the diagram or not?
How about the good old-fashioned pointer?
Or a pointer, if you can reach. I think we've got a pointer right next to the entrance to the... In fact, Dr. Cotton, did you bring one of these membranes to court today?
I gave it to you, and presumably you brought it to court.
At this point, I hope I did. All right, Your Honor. I'd ask to have marked as people's next in order, I believe, 245.
244, I believe. I think it is 244. Mrs. Robertson, 244? 245, I stand corrected.
And I have provided the defense one of these items as well.
All right, proceed.
Dr. Cotton, if I can show you what seems to be a object about the size of what, 8 inches by 8 inches or so?
Yes, it's actually 20 centimeters by 20 centimeters.
Okay, that sounds like a more precise description. What is, and referring you to what will be marked People's Exhibit 245, what is that?
This is how the membrane comes to us. It's in these red pieces of, this is just a piece of paper, and it has written on it MSI, and that's the manufacturer of the membrane that we use. And sandwiched in between the two pieces of paper is a nylon membrane. Now, if I were doing this in the lab, I wouldn't be touching it with my fingers. You would be handling it with gloves because fingerprints wouldn't be helpful. Any of the oil from your fingers is really a problem. And this hasn't been used in the lab, so it's clean. And this is the nylon membrane. So in the example here, you have a sponge, and in our lab we use something that's called a wick, but it's all going to do the same thing, so even though this is slightly different from my lab, I'm just going to stick with this. And then on top of this, and the sponge is soaked with some solution, and on top of the sponge you place the gel. So you can see the gel here is green, and you have a green layer here, that's the gel. Of course, the gel isn't really green, it's really kind of just whitish. And you can see that the membrane isn't really orange, but they made it orange. And anyway, so you have that gel, and you simply, if I had a gel here, I'll use the top of this thing, you would very carefully take this nylon membrane and lay it down.
Actually, I'm sorry, Dr. Cotton, perhaps you could demonstrate that on the actual bar in front of the jury so that more can see.
I had my gel sitting right here. I would carefully lay this nylon membrane down on top of the gel and let it fall down easily. And it would be wet beforehand, and the gel is damp. And then you can, when I did this in research, I used my fingers, but in the lab we would now use a pipette. But you would smooth down the nylon membrane so that it was in easy, close contact with the gel.
All right, Dr. Cotton, could you keep your voice up, please?
Yes, sir, thank you. Now, the purpose of this is to have the DNA move out of the gel and come in contact with the nylon membrane. So if we go back to the diagram here, the salt solution that's in the sponge will move up through, let me finish the order here. We have the sponge, the gel, the membrane, a big stack of paper towels, and a weight. So the idea is that the solution moves up, that's soaking in the sponge, moves up through the gel and carries with it the DNA. And it's sucked up, the solution is sucked up into the paper towels, but the DNA becomes trapped on the nylon membrane. So if you were to go back to this diagram, the membrane would be layered right on top of the gel, and the DNA would move up through the gel and become attached to the nylon membrane. The paper towels and the weights serve the obvious purpose, the same as if you had a puddle on the counter and you put the paper towels on it, the solution would be soaked up into the paper towels. It's not a very elegant contraption, but this is one of the most important procedures in all of molecular biology, and it's used all over the world.
Mr. Clarke, the diagram that Dr. Cotton referred to, again, the drawing, I believe that's the electrophoresis diagram.
Correct, 244.
Now, I have omitted one important feature here, and somehow I always have trouble getting this in the right order. Just before you set this setup up, you treat your gel so the DNA fragments that are double-stranded will come apart and become single-stranded. And you do this by soaking the gel in a solution of sodium hydroxide. So basically, the other method that we talked about was heating it, but if you heat it, the gel will just melt, so you can't do that. So you're going to separate what was double-stranded to now be single-stranded in the gel, and then you're going to do this transfer so that as the DNA is bound to that nylon, it's in a single-stranded form. And that's the important thing that I left out.
Actually, two questions if I can. First of all, why doesn't the DNA travel through the membrane and up into the paper towels?
The DNA binds to the membrane and it becomes trapped there. The membrane doesn't have sufficiently large pores in it for the DNA to get through.
Okay, and then you also referred to this process as not being particularly elegant, is that right?
Well, it's very elegant, but it's not very fancy. It doesn't require a lot of equipment. You don't need any electricity. You could set it up in your kitchen if you really wanted to.
Is this particular step limited to forensic science, or is it applicable, is it actually used in any technique involving use of the RFLP method?
It's used in any technique where RFLP is used, and it has other uses as well. But if you go into any of the scientific journals, you probably can't go through a journal that talks about molecular biology or biochemistry without finding an article where this procedure is used. This procedure is called a southern blot, and it doesn't have that title. Oh yes, it does have the title here on the diagram where it says by a technique known as southern blotting. It's called a southern blot because the scientist who devised this procedure in the late 1970s, mid 1970s, his name is Ed Southern. He's from England, and his last name became attached to the procedure because it's so widely used. So it's called a southern blot. That's the name of the scientist who devised the procedure, and it's been used since that time.
All right. So now is it correct then that this membrane will be marked People's Exhibit 245 in this process now has single-stranded DNA attached to it?
Yes, and the important thing is, however the strands were separated in the gel, they are now, that is, however the DNA fragments, that is the large one versus the small one, were separated in the gel, that same spatial separation is now on the nylon membrane. So the DNA is attached to the same nylon membrane, and the pieces are in the same relative positions that they were in the gel when the gel was turned off.
What's the next step in this RFLP typing process after you have the membrane with the DNA attached to it?
The next step, I have to read what's on the board for a
All
second
right.
to see where I am on this. Okay, so over here, after the blot, you have this statement on the board, which is correct. The DNA fragments are now on the nylon membrane, but are not yet visible. Now, actually, it shows you as if something was visible, but really, when you put the nylon down and you take the nylon off the blot, it looks just plain, just like you saw it. It doesn't, except it's wet. And now you get to the point of the next few steps that allow visualization. And here's where it becomes important that you understand that you can take DNA fragments apart and put them back together, because now you're going to take what's called a DNA probe, which is simply a piece of DNA that you've made in the laboratory and has a radioactive tag on it. It's single-stranded, and if your repeat was CAT, your probe would be GTA.
Actually, you used an earlier drawing that demonstrated a tag, did you not?
I did.
Perhaps you can refer back to that. And Your Honor, that would be People's Exhibit 239, labeled single-stranded DNA.
So, in my single-stranded DNA drawing, I didn't draw the repeat CAT, but let's suppose this is our repeat. If this is our repeat, the DNA that's on the nylon is now single-stranded, so you have this available to collect the second strand, and you would have the other side available to collect a second strand. So you take the nylon membrane and put it in a plastic tub, or you can put it in a tube-like thing, but I'm going to use the example for a tub because it's a little easier. We used in the lab a Tupperware container, a square one, in which that was just about the size of that nylon membrane, so the membrane would be flat in the bottom. And then the membrane is covered with a salt solution. The radioactive DNA probe is added to that solution, and that solution is let sit at a specific temperature overnight. So overnight being, about 16 hours perhaps. And during this time, this radioactive probe DNA, which is simply, the probe is actually, for what we do in the lab, attached to C's and G's. During this time, this probe that you've added will zip up with any DNA on the membrane that it can appropriately zip up with. The radioactivity is something that you can follow. If you take your membrane, add your probe, let it go overnight, take your membrane out, rinse off the excess probe. If I held a Geiger counter up to it, I would get a few clicks, the Geiger counter being able to detect the radioactivity. But that's not accurate enough. So instead of a Geiger counter, you would then, if we can go back here, here's our membrane that now has some probe bound to it. To detect where that probe has bound, you lay an X-ray film over the membrane. You do this in the dark. You put it in a metal cassette that sort of looks like two book covers that then close up together. And the radioactivity that's on the membrane will expose the silver grains on the X-ray film, creating a dark spot or a line. It's really a line. And that is the image of where the probe bound to the DNA. So if we go way back, if we go back to this diagram, this was originally our gel. Now this pattern of DNA is on the membrane. And now you've added a probe to it, and the probe has bound specifically. We make the probe so it binds specifically to the CAT repeat. That's what it's going to do. So the probe is bound specifically here and here. And it's been washed off everywhere else because only in these two positions is there a piece of DNA that it can come back together with. Then we've laid a piece of X-ray film over this membrane and allowed it to expose. And on the X-ray film, a dark line will appear in these two relative positions. That dark line tells you where the DNA fragment went to in the gel when the gel separation of these pieces occurred.
All right, referring to People's Exhibit 244, Your Honor, the drawing.
Yes.
Dr. Cotton, these probes then, are they specifically designed to look for that repeat sequence that you're searching for on the membrane itself?
Yes.
And when they recognize the sequence that they're looking for, the probe then actually mates up or zips together with the DNA that's already on the membrane from this blotting procedure?
Yes.
Is the probe then simply a means for you to be able to see where the fragment is on the gel that you're actually looking for?
Yes. The probe, this is your tracer, it's your eyes, it's your tag. And you sort of, we talk about it like it was a thinking thing, but of course it's just a piece of DNA, and this is just a chemical reaction that allows these two DNA strands that were single-stranded to come together. So it's simply your way in the laboratory of identifying where these pieces of DNA went that had the repeat that matches to the probe.
Then is the use of this radioactivity and X-ray film so that you can actually look and see where these fragments are?
Yes, that's exactly what you're doing.
And is that done by your own eyes?
When you look at the X-ray film, yes, you're looking at it.
Does the X-ray film have a particular name in the RFLP testing process?
It's properly called an Autoradiograph. In the lab, we refer to it as an Autorad. It's just simpler to say, and that's what most people would that's the term most people would use.
All right, Your Honor, at this point I'd like to have marked as people's next in order.
Is that 246? All right, People's 246, Mr. Clarke.
Yes, a single, what I believe to be an autoradiograph that's been, a copy which has been previously provided to counsel.
All right, single autorad.
Dr. Cotton, just showing you what will be marked People's Exhibit, I'm sorry, 246, Your Honor?
246.
What is that?
This is a copy of one of the autoradiographs that was produced in this case.
And is that typically what an X-ray or autoradiograph looks like following the use of this RFLP typing process?
Yes.
All right, without getting into what the samples are on that particular test, and with the court's permission, could Dr. Cotton simply hold that up so that the jury can see it from a
Yes.
distance?
This is one of the film, well, this is a copy, but the copies are very good, they don't, so that if I,
Objection on this is not a response to the question.
Sustained. Mr.
All right.
Clarke.
All right, Dr. Cotton, if you would just hold it up at the moment, and we will return to this particular autoradiograph later. But is this typical of what such an X-ray looks like at the end of this RFLP procedure?
yes, it is,
Okay, and in
without
fact,
all the lettering.
okay.
You have to subtract out the lettering here and the lettering down here.
The lettering you're referring to at both the top and bottom of this particular exhibit, is that done to aid in knowing what sample is, which sample is which, basically?
Yes.
Okay. All right, fine. Would that autoradiograph then represent what is labeled on the RFLP method chart at step 7 and then step 8?
It would only represent what's labeled in the step 8.
Okay. So step 7 is describing the creation of this X-ray film in its early stages by laying it on top of the membrane.
Yes.
All right, very good. Perhaps I can take that back. And, if you would, could you have a seat back on the witness stand?
And Mr. Fairlow, can you rescue the diagram easel there, please?
Actually, with respect to the RFLP method, the large chart, could that also be brought down so that all of the jurors could have a look
Yes.
at it?
Fairlow, Mr. Clarke, perhaps you should both try to handle that. Yes. All right, thank you, counsel.
Dr. Cotton, is that then a capsule summary of this RFLP typing approach?
It is.
You are familiar with the term controls.
Yes.
What is a control?
Control is usually some kind of sample that helps you to determine whether all the procedures that you just finished using worked as they should. And it usually is a sample that you know what the results from the control should look like, and if they look appropriately, then it gives you some information that your test was done appropriately.
Now, referring to this RFLP typing process, do you utilize controls as part of that testing?
Yes, we do.
Are there a number of different types of controls?
Yes.
How do you know when a control isn't working properly?
Well, it depends on what kind of control you're using, but for this procedure, you are ultimately looking at that X-ray film and visualizing your result, and later on you might make some measurements, but if we just talk about looking at it for the time being, the controls have a very typical appearance, one that you are accustomed to seeing. So if they look as they should, then you would make a general first impression assessment that the test and the procedures that you used had been done appropriately.
What if the controls didn't work properly in a particular test?
Generally, if they haven't worked properly, you would either go back and repeat it if there was a possibility to do that, or if you couldn't repeat it, you would probably just say that your results are inconclusive, because if your controls haven't worked, there's some implication there that how you worked on the actual samples may not have worked either.
Now, this autorad in People's Exhibit 246 represents an autorad from an RFLP test, is that right?
It does.
This autoradiograph, is it in fact preservable? In other words, is it in a form that, for instance, can be looked at a later time or by other people?
Yes, the autoradiograph is your permanent record of the results of the test.
As far as the RFLP typing results, and first of all, did your laboratory obtain RFLP typing results in this case?
Yes, we did.
Are those RFLP testing results reflected on autoradiographs, as an example of which is People's Exhibit 246?
Yes, they are.
Now, I'd like to discuss a different area with you, and it relates in particular to when you are reading results from an autoradiograph. First of all, do you do that?
Yes.
And you look for certain things, is that right?
Yes.
If I use the term match, what does that mean to you?
It means to me that on the autoradiograph, the position of 2 DNA bands, or possibly the positions of all the bands from one sample to another, are so close as to be visually that I couldn't distinguish one from the other.
What does that mean, that you can't distinguish one from the other?
They look alike.
Does that have a particular meaning in this RFLP typing process?
Of course.
What's that?
From person to person, there's so much variation in the length of the fragments that have a repeat that if you look at several genetic locations, and for each genetic location 2 samples are the same, and you look at another and they're the same there, and you look at another and they're the same there, and you keep doing that, the more genetic locations at which 2 samples are the same, the higher the likelihood that the 2 samples are in fact from the same person.
All right. Your Honor, if I may, return to People's Exhibit 242, the large board, because I believe it has an item that I'd like to ask the witness another question about. Put it away too quickly. Thank you. Dr. Cotton, referring you to what would be step 8 on this board, People's Exhibit 242, does that particular depiction, would that help you in describing what you mean by a match?
Yes.
All right. Could you then go ahead and describe that? And if the pointer would help,
Okay.
please feel free.
This diagram and the things that I drew on the chart are illustrating the kinds of results you would get when you looked at one location on one chromosome. So since you get you get 2 copies of the chromosome, you get one piece of information that you inherited from your mother and the other from your father. And so if we look at this sample that would be basically on your left, you have 2 dark lines on the Autorad indicating the 2 DNA bands that are from this sample. And then if you go on to the middle sample, you have 2 bands on this Autorad from this middle sample. And then if you go to the third sample, you also have 2 bands. The middle sample and the sample on the left that I'm pointing to, the bands are in essentially the same position. So it is possible that sample number 1 and this middle sample, sample number 2, could be from the same person because they have DNA bands that are in the same position. This third sample on the right, which also has 2 DNA bands, these bands are clearly in a different position than either the first or the second sample. That tells you immediately that the DNA in this third sample must be from a person who is different from either of these first 2. These first 2 could be from the same person, but this third one must be from a different person because the DNA fragments are in different positions, which means they're different sizes.
Would that be an example of an exclusion?
If you were comparing, suppose this was a known individual and these were 2 evidence samples. This known individual in the third lane over would be excluded as being a possible contributor to the 2 evidence samples in the left lane and in the middle lane.
This is done visually as a result of the autoradiograph produced at the end of the RFLP typing process?
It's done visually, and that's followed up by a more sophisticated measurement. But in fact, the first thing that you do when you get your X-ray film off is you look at it. And you look at the controls, and you look at the samples on there, and you make some immediate judgment about whether it appears that the procedures worked okay and whether it appears that you have inclusions or exclusions, and based on those inclusions or exclusions, then you might do further testing or you might not.
Following this visual determination, do you then in some instances turn to another process basically to determine whether or not your visual opinion is a correct one as to a match?
Yes.
All right. We'll return to that later. But what I'd like to turn your attention to, Your Honor, I'm going to leave the chart because I don't want to have to ask to bring it up again. And that relates to the amounts of DNA that are in a sample. And I'd like to focus your attention again on this RFLP typing method. If there's no DNA in a particular sample, what do you see at the end of the process on the X-ray or autoradiograph?
If there's no human DNA in a particular sample, at the end, you don't see anything. The lane is blank. There are no bands. It's just empty.
Are there other types of DNA that you could see?
For the procedure as it's used in laboratories for human identification testing, you're specifically looking at, you want to be looking at human DNA, and the test is designed to only detect human DNA. And therefore, if you were using it in a research lab, you could see other types of DNA. But for this purpose, you're only looking for human DNA. If you have no human DNA there, you won't see anything.
So in your laboratory, your interest is in human DNA as opposed to looking at, for instance, animal DNA or other types.
That's right.
What if you have some DNA in a sample? Would you necessarily see results at the end?
If you had some DNA in a sample, you might see some results at the end. There are 2 measures of whether or not you might see results, and one is quantity and the other is quality.
What do you mean by that distinction?
If your DNA is in good condition, and let's just go back to the spool of thread analogy, if your DNA is unwound off the spool and it's all in one piece, that's good condition. It's like it was in a nice fresh cell that somebody just drew out of your arm. If your DNA is in good condition and you have enough of it, you will get a result. If your DNA is broken up in a random manner, and the proper term for that is that it may be degraded, you may or may not get a result. And when you think about DNA that's broken up or degraded, you have to think of it sort of as a continuum. It could be only slightly degraded, that is, it's only broken up a little bit, in which case it won't affect your result, you will get a result. Or it can be very, very broken up in very, very small pieces. And if that was the case, you would not be able to get a result with this RFLP test.
What happens with DNA? Does it in fact die or degrade or perish?
Well, I wouldn't use the term die or perish. It doesn't really fit. Degrade is the right term. Or if you want to make that simple, you could just say it's become broken up into many pieces. And that can happen as a result of environmental effects. And you can generally sort of assume that moisture and heat facilitates degradation. That is, it occurs more rapidly, and cold and dry conditions preserve DNA.
If DNA suffers this degradation process, will that result in changing the types from the original type of the sample to a new and different type as a result of the process of degradation?
No.
I'm sorry?
It will not.
So this process of degradation, can it change my DNA into looking like your DNA?
No.
Or your DNA into looking like the court's DNA?
No.
Or any members of the jury or the audience?
No.
Taking you back to this small amount of DNA, what if you have more DNA in a better condition, let's say? Then what do you see?
If you have more DNA and it's in good condition, then you will see bands on your autoradiograph or your Autorad, and each time you use a different probe, you'll see bands that probe recognizes, so you can get a lot of information if you have a good amount of DNA and it's in good condition.
Where do you draw the line? In other words, how do you know there's enough DNA there to be able to make an opinion or render an opinion about results from a particular test?
Well, you can sort of go about it from two directions. You can measure how much human DNA you have before you do the test, and you can run what's commonly referred to as a mini gel, which is a really small gel, about 2 inches by 3 inches, and assess whether the DNA appears to be broken up. So you can measure its quantity and you can get an estimate of its quality. And you could do that before you did the test and then say, well, it looks good, let's do an RFLP test.
Is that, by the way, using the same process of electrophoresis for this mini gel?
Yes, exactly the same.
And that's simply a step to help you evaluate, that is to determine approximately how much DNA is in a sample before you test it?
Yes. If you go back to this green depiction of a gel,
At step 3 on People's 242?
right, if you just made that a lot smaller, and which would then represent your mini gel, you can stain that gel with a dye in which you get a pink smear that looks like these smears that are on this diagram for step 3. And if you had a smear like what's shown in step 3, you'd look at that and you say, well, gosh, I've got DNA sort of spread out all the way from the top of my mini gel down to the bottom. that's pretty degraded, and if it looked like that, you'd be sort of, you wouldn't be sure whether it would be good enough for RFLP or not. If this diagram in step 3 had only shown the pink color at the top, that's sort of what it would look like if it was in good condition and you ran a mini gel. You're looking at a huge piece because you haven't, when you do this mini gel, you haven't cut it with your restriction enzyme yet. So actually, if we could insert an extra step here to be 1A, so that you extracted your DNA and then you ran your little gel and then you made this judgment. If you had DNA that was just hung up at the top of the gel, that would say, you'd say, well, I have very good DNA here. I should be able to go on and do an RFLP test. If the DNA is all spread out such that it looks relatively degraded, then you would have to make a judgment about whether or not you thought it would be good enough to do an RFLP test or you were going to do some other kind of test with it.
As far as these bands as you see them, and referring to, for instance, step 8 on People's Exhibit 242, those are fairly dark bands in that depiction, correct?
Yes.
You've also held up one autoradiograph, People's Exhibit 246, which also shows a number of bands that seem to be very dark. Is that right?
Yes.
In fact, actually, if you could, with the court's permission, could the witness hold 246 up again?
Yes.
First of all, Dr. Cotton, to your knowledge, will we return to autorads and actually show them in closer detail to the jury?
Yes, we will.
Okay, but just for the moment, and if you could hold that up. I don't know if you can hold it up without that white background. Let's do that.
Yeah.
Why don't you have Dr. Cotton just step down over there?
All right, that's fine.
She can just hold it up against the white background.
Dr. Cotton, again, showing People's Exhibit 246. That shows a number of very dark patterns. Is that right?
Yes,
Or bands, is that the appropriate term?
it is.
Now, with regard to samples, for instance, that you receive in your laboratory, will the darkness or the intensity of those bands vary?
Yes.
Why?
The darker the band, that reflects you've had more probe bound to it. More probe means that you had more DNA there. So if we just, we don't have anything on here that has very light bands, but if you had something that had very light bands, that would be an indication that you didn't have very much DNA. If you have bands that are darker, that means you have more DNA. Now, there's other ways you can make the length of time that you lay the X-ray film over the nylon, the longer you lay it on there, the darker everything becomes, but it will still be relative. A darker band generally has more DNA than a lighter band. And there's a few reasons why that, there's a few technical exceptions to that, but that's the correct generalization.
All right. Then if you would have a seat back on the witness stand. Is it the case then, Dr. Cotton, that with regard to samples with not much DNA in them, but enough to produce a banding pattern, you may see bands that are faint?
That's exactly right.
Is faint a term that you use in your laboratory?
Yes.
What does it mean?
It means that instead of a dark, crisp line, you have a difference between where there's no band and where there's a band, but the level of darkness there, that is the intensity, is much less. It's light.
Does that have any significance in your interpretation of results?
You are always looking at the position of the band. So whether it's dark or it's light, it's the position that you're interested in. it does affect, that is, you think about it in your interpretation because you're assessing, I have a lot of DNA here, I don't have very much here, and you may use that information in your interpretation, but it's mainly the position that you're interested in looking at.
What about in terms of faint bands and whether or not you can see them? Does that play a role? I assume that plays a role in your interpretation.
Well, if you want to interpret them, you better be able to see them.
Are there any policies in effect in the laboratory at interpreting, for instance, results that have faint bands?
I don't believe that we have a specific written policy. We do have a common practice in our lab in dealing with faint bands, and we do follow that practice generally whenever we encounter faint bands. It's not actually a written part of our procedure.
And what is that procedure, though not written?
There are two parts to it. In the further analysis that we haven't really discussed yet, which involves a computer imaging step, there is a standard level of darkness that we ask that the computer see in order to call something a band. In addition to that, we have to be able to see it ourselves. That is, both the computer has to be able to see it and the analyst has to be able to see it. And if we really are struggling with whether or not a band is light, we will ask several people to come in and cover up the adjacent lanes and make an assessment as to whether all the people involved in doing the analysis of the test agree that they can see a particular band.
When you say cover up the adjacent lanes, is that to ensure that whoever's looking at it wouldn't be biased by the presence of bands in other samples?
That's right. If I'm working on something or another PhD staff or one of the analysts is working on it, if I'm not sure whether or not something is intense enough for me to use it further in the interpretation, I might lay a piece of cardboard down on either side and just say to one of my coworkers, would you come in and look at this and tell me where you see bands in this lane? And we don't always do that, but that's generally what we do when they get quite light.
As far as this process of DNA, once it's degrading, does this degradation process ever lead to bands appearing that weren't in the original DNA in the sample?
No, it doesn't.
So in other words, you either see the DNA from the original sample, or if the degradation is too great, you don't see anything at some point.
Well, you've just said what the extremes are. You may have an intermediate result where you have the DNA is somewhat degraded, and the effect that that has is you tend to lose the bands that are larger, that is at the top of the gel. So you may end up with a partial set of bands. You don't see the ones that are large, but you see the ones that are smaller in size. And that can happen also. What won't happen is that with degradation, you won't have a band at one size and then have it converted to now a new band at a different size. That's not going to happen.
Okay, and perhaps lastly for today, with regard to this computer imaging process that you just described, can you tell us more about what that is?
It's sort of like this Elmo machine, actually. Well, kind of. The computer imaging system consists of a light box and on which you lay your film, a camera at the top, and a computer monitor so that the camera is capturing the image of the autorad and displaying it on the computer screen for you. And then you're asking the computer to help you do some manipulations in terms of saying, here's a band, and here's a band, and here's a band. And then the computer will also, because of its software, do a calculation for you that says the band in this position is approximately 5,000 base pairs.
Now, when you say the autorad that you have the camera actually focus on, is that an autorad just like People's Exhibit 246?
Yes.
And then this camera and this software in combination then review the results on that particular autorad?
Well, the reviewing makes it sound like it's thinking about it, and it's not. What it's doing is visualizing. It's a video imaging system. It's taking a picture of that autorad and displaying it on the computer monitor, and then you are directing the machine to do particular things, not to the film itself, but to the image.
And what are those things that you direct it to do?
You're telling it where the control lanes are, you're telling it where the sample lanes are, and you're telling it which lanes you want it to do a calculation to find a band. That is, the computer will basically scan down, and where there's a dark area, it will identify that as a band, and where there's, so it's looking for changes in levels of darkness. So if it's clear and then it sees a dark area, then it identifies that as a band, and then it sort of scans down and finds another band and identifies that. So it's making an identification of a band position, and it can do this correctly and it can do it wrong, and the operator has to help it along. And then once you're happy that it's identified bands and you agree that those are bands, it will do this calculation for you that will give you an estimate of the size of that DNA in base pairs.
And that's the purpose of this imaging process, to assist you in interpreting results, is that right?
That's right.
And also to establish these sizes for the various bands according to the methods that you just described?
That's right.
All right.
Good spot?
Yes, thank you, Your Honor.
All right. All right, ladies and gentlemen, we're going to take our recess for the afternoon. Please remember all of my admonitions to you. Don't discuss the case amongst yourselves. Don't form any opinions about the case. Do not conduct any deliberations till the matter has been submitted to you. And do not allow anybody to communicate with you with regard to the case. As far as the jury is concerned, we'll stand in recess until 9 o'clock. And we have a hearing tomorrow morning set at 8 with Mr. Ullman and I don't recollect who from the prosecution. Photographs tomorrow, 8 o'clock. All right, we'll stand in recess. And Dr. Cotton tomorrow morning, 8:45. All right, thank you. We'll stand in recess.